Back in 2001, something incredible happened. For the very first time, humanity got to read its own instruction book. An international team of scientists published the first draft of the human genome. This was a monumental achievement, a moment that unlocked this extraordinary trove of information about who we are. But how did they even start to read this book? Well, it was a historic scientific quest truly unlike any other. So, what exactly is in that book? Before this project, that was one of the biggest mysteries in all of biology. Scientists knew our DNA held the code, sure, but they really had no idea of its sheer scale or what secrets it was hiding. They were basically setting out to map a completely unknown territory. And what a territory it turned out to be. Our genetic code, our genome, is made up of 3 billion letters. To put that in perspective, if you printed that out in regular font size, you'd have a stack of books 200 feet tall. I mean, just imagine that. That was the scale of the challenge facing the Human Genome Project. So when the results finally came in, the story they found, well, it was not at all what scientists were expecting. The draft sequence revealed this genomic landscape that was strange and beautiful and way more complex than anyone had ever imagined. It fundamentally changed how we understand ourselves. So how on earth did they do it? How do you even begin to read a 3 billion letter book written in a language you're only just starting to figure out? Well, it took an absolutely ingenious strategy and a level of global cooperation that had never been seen before. Okay, so the strategy they used has this super technical name, hierarchical shotgun sequencing. But the book puzzle analogy is just perfect. Think about it. You can't read a 3 billion letter book all at once. So you shred a bunch of copies into tiny, manageable pieces, read those little snippets, and then use the parts that overlap to figure out how to put it all back together. So here's how that puzzle solving actually worked, step by step. First, they used a kind of fingerprint map to get large chunks of DNA in the right general order. Then, they took each of those big chunks and shredded them again, this time into small, readable bits. They sequenced those tiny fragments, and then, finally, they used some seriously powerful computers to stitch it all back together into one continuous draft sequence. And you gotta understand, this wasn't just one lab somewhere. This was a massive global effort. We're talking 20 centers spread across 6 different countries, all working together. And the DNA itself, it came from a diverse group of anonymous volunteers. The process was so strict that not even the donors themselves know if their DNA was part of it. It was truly a genome for all of humanity. And when you look at the actual data, the sheer scale of that collaboration is just staggering. You can see the insane volume of sequence that was produced by the top centers, with the Whitehead Institute at MIT, the Sanger Center in the UK, and Washington University really leading the charge. Every single one of these numbers represents a colossal amount of work. All this collaboration, it built up to this incredible pace. At its absolute peak, the project was sequencing over 1,000 letters of DNA code every single second, 24/7. It was just this fire hose of data, a pace of discovery the world had literally never seen before. All right, so with the map finally in hand, scientists could start exploring this brand new world. And what they found, well, it was shocking. The neat, orderly genome they thought they'd find turned out to be this wild and surprising landscape, just full of unexpected features. Okay, here's the first huge shocker. For years, scientists had predicted humans would have something like 100,000 genes. Makes sense, right? We're complex creatures. But the draft sequence, it showed the number was actually closer to 32,000. We had barely more genes than a worm. So the big takeaway here is, our complexity doesn't just come from how many genes we have. The next big surprise was about, well, everything else. For decades, the 98% of our DNA that doesn't actually make proteins was just dismissed. They literally called it junk DNA. This project completely flipped that idea on its head. Turns out, it wasn't junk at all. It was a treasure trove. And you can really see the real estate breakdown of the genome here. Just look at that tiny little sliver for protein coding genes. It's like 2%. The huge majority is made up of all these other things, many of them these repetitive bits of code with names like lines and signs. This is the so-called junk that turned out to be so incredibly important. And this quote from the original scientific paper just says it all perfectly. This junk DNA wasn't just filler. It was a fossil record, right there inside our cells. It's a history book of our evolution, recording ancient viruses, huge genetic events, and all the forces that shaped us over millions of years. And by studying the tiny changes, the mutations in these repeats on the X and Y chromosomes, scientists could use them like a molecular clock. And one of the first really cool things they found was solid proof that the mutation rate in sperm is way higher than in eggs, a fundamental secret of our own biology, just revealed by digging through our evolutionary history. Okay, so beyond all the fascinating biology, the most immediate real-world impact of the Genome Project was in medicine. It basically gave doctors and researchers a whole new toolkit for understanding and fighting human disease. So before 2001, if you wanted to find the single gene responsible for hereditary disease, you were in for a long, long journey. The process was called positional cloning, and it was kind of like trying to find one specific house in a giant city, but with no map, no street signs, and no addresses. It could take years of just painstaking work. The genome sequence changed everything. It was the ultimate map. Suddenly, researchers could figure out the general genetic neighborhood of a disease and then just look up the address. They could scan the sequence and find the culprit gene in a tiny fraction of the time. And the results came fast, immediately. Using this new draft sequence, scientists started knocking down disease genes left and right. From breast cancer susceptibility to hereditary deafness, the map just accelerated discovery across the board. This gave real hope to countless families. It also gave us, for the first time, a complete parts list for the human body. So researchers could search the sequence for genes that looked similar to ones they already knew how to target with drugs. And bam, they uncovered brand new players in major diseases, like a new serotonin receptor that's crucial for mood disorders, or a new enzyme linked to Alzheimer's. So the big takeaway here is this. It wasn't just about finding what was broken. It was about discovering totally new ways to fix things. The number of potential drug targets just exploded, from maybe a few hundred to thousands, opening up entirely new ways to think about developing medicines. But this first draft, as amazing and revolutionary as it was really just that, a draft. It was a starting point. And honestly, it probably raised even more questions than it answered, opening up these huge new territories for us to explore. So what other secrets are still written in our code, just waiting for us to find them? The authors of that original paper ended with this absolutely beautiful quote from T.S. Eliot. And it's just the perfect way to think about this whole thing. The Human Genome Project brought us all the way back to the very beginning, our own biological code. But it allowed us to see it and to see ourselves with completely new eyes, to truly know the place for the first time.